primarily primarily in the area of adaptive design and statistical issue in precision medicine. She has authored more than 40 articles in peer-reviewed journals on methodology, study design, and data analysis and reporting. And Sami is also a co-founder of DaShu, that is a non-profit organization founded to promote research and education of 12,000 members. She is leading the team in DIA Innovative Design Scientific Working Group of Oncology Drug Development and a Small Population Working Group for Rare Disease. She is also an active member in ASA and ICSA to serve the biostat and data science professional community. So our another speaker today is Dr. Rong Liu, who is currently a senior director, head of early clinical development biostat of hematology and cell therapy biostat in BMS. Prior to joining BMS, Dr. Liu was the director of oncology stats at Bayer. She had more than 15 years of experience in design and analysis of clinical trial for early and late development. After receiving her PhD from Virginia Commonwealth University, Dr. Rong has accumulated extensive industry experience from leading pharmaceuticals such as Merck, Bayer, and BMS in various disease areas. Dr. Liu is currently the co-lead of combination therapy sub-team in ASA Biopharmaceutical Scientific Working Group and the editorial board member of Contemporary Clinical Trial and a member of Consolidated Standard of Reporting Trial Extension Group, developing of a reporting guidance to improve knowledge transfer. Another speaker, or the main speaker of today, is Chen-Kun Wang, who is Associate Director in Biostat Department at Vertex Pharmaceuticals. She joined Vertex in 2015 after receiving her PhD in Biostat from Indiana University. He has over 4 years experience in the rare disease of cystic fibrosis, from first in human study to NDA submission. In the recent 2 years, he is working in a novel cell therapy for type 1 diabetes at lead station. Chen-Kun is also a group member of the BI Adaptive Design Working Group on historical borrowing. So with all those introductions, so I'll give the floor to Chen-Kun. Maybe Chen-Kun, you can show your screen and start your presentation. Maybe before that, for the audience, if you have any question, you can put your question in the chat box, or we are going to unmute everyone at the end of the presentation, so you can also vocal your comments or question at the end of presentation as well.
Okay. So thank you, Zhaoyang, for the good introduction. I didn't know you're going to read all our bios already. I think that's very well said for all the speakers, and I'm very glad to be part of the presentation. My role is very simple. I'm really helping to open up this presentation and to really, on behalf of DIA NEED Working Group. So today's topic is really about using external data, and this working group has been working on this together for quite some years. If you go to the next page, yes, we have quite some members representing industry, representing academic. Our purpose is really specifically focusing on how to generate evidence or needed evidence for rare disease. That's why it's called a NEED subteam. And the NEED subteam is part of the DIA ADSWG, Adaptive Design Working Group previously. And I'm also part of the leadership with this working group. So that working group is very large. We have over about 300 colleagues from the whole industry, either US or Europe overall, working on different topics. And last year, we changed the name to IDSWG, Innovative Design Scientific Working Group, which means is anything talking related to innovative design would be part of our topics covered. So feel free to reach out to me if you're interested to join this working group. We have conducted a master protocol and CID working shop even a year ago, and this year, November, We will have another one working with DIA, have a master protocol and CID program in November. It's coming also soon with a lot of great speakers invited. If you go to the next page. So this NEED working group sub-team within the IDSWG has been working on this topic specifically and for the last 2 to 3 years. And what we initially thinking is in rare disease, there's a lot of need for external information generated to be utilized part of the clinical trials that really would help improve, the information we are really hard to get in a rare disease setting. So our initial thinking is how can we have a very handy reference to get? If you go back to the last page, Chen-Kun. How can we have very handy things we can get reference if we want to do a historical control? What information we get, what kind of design we should do, what type of a methodology analysis we can utilize in the conduct of study. And even if there's anyone have approval cases in using external data as historical control with a successful story as proved by the regulatory or maybe failed story. We want it all as a framework and provide a practical roadbook or as a handy reference to the members in the industry. And actually that worked very nicely. We use a lot of effort to do this research and collect those information and recommendation. So all of those has been published in 2020 last year in the Medical Journal of Orphan Rare Disease. I think people really find this very helpful. So today we're going to talk about some elements from that paper. If you go to the next page, we can quickly talk about what's historical control, but I don't think it's a new concept. If you look at old days, when drug development is not very well established, the doctor would look at the patient, how better they are with a different treatment. It's your self-control. Or looking at a few of the patients, a group of patients to the similar situation treated in different manner, right? So that's considered old days. Of course, when the statistician comes in, the pharmaceutical industry agency really well established, we comes to the era of a gold standard of randomized clinical trials. So definitely it's very important scientifically and really remove the potential bias and provided the compatible groups to come to the conclusion of scientific questions. But it is getting very difficult for the rare rare disease drug. So in 1983, there's Orphan Drug Act released by US government. Since then, due to the exclusivity and the patent extension, reduction of the tax credit, the pharmaceutical company really gained a lot for doing this, and more rare disease drug has been approved since then. So we are accessible to a massive historical data, not only rare disease per se, actually due to the technical industry changes, a lot of information in the real world setting are gathered information might help the clinical trials. So it's getting more and more important how we reconsider the historical control as the new concept to be part of the clinical trial design. If you go to the next page, there are a few important topics in our paper beyond the case studies, which Chen-Kun would introduce later on, and the methodology. But the framework overall is we want to help the community, whoever are thinking about using historical control, to talk about, for example, what's the source of your historical control? We can think a lot, medical charts and claims data, or registry data, natural history data, or even clinical trials. If you have the same indication disease, smaller trials, or from the previous other companies' trials in a similar competitive setting in the same indication, might also could be utilized as external information. But the data formatting and the quality of data is definitely a problem. So should we use historical control? If we go to the next page, yeah. So we need to think through quite some questions. To me, two very important questions to think about is, first of all, whether it is ethical to treat your patients in the control arm, or even it's enough to enroll patients in your control arm. Another question also important is whether you have a high quality data in your indication you are investigating. If not, maybe that's also a moot question to move forward historical control. So when those questions are clearly answered, then you can think about if it's in a regulatory setting for confirmatory trial or be utilized as part of the NDA filing or BLA filing. That also related to how much effort you need to pre-plan closely with regulatory agencies. But to me, overall, if it's unethical, you really cannot do historical dual control arm or even not enrolling NASH patients. You either could do historical control, you can do what we call synthetic control. Either way, to utilize external information to be part of the clinical trial design. This will be definitely very efficient and benefiting to the patients too. But there are a lot of disadvantage also using historical control. If you go to the next page, nothing is for free lunch, as we all know. So this shouldn't be a first time people thinking about this. You know, it's not a randomized trial. The data quality is all over the place. It really depends on what type of external information you gathered, either from natural history, registry trial, or even from previous clinical data. Maybe that will be slightly better. Data collection formatting could be different. You think about the different schedule assessment, it could be even different. That might impact your primary endpoint, even definition. Simple example, for example, like in oncology, some rare disease in oncology, PFS, if your schedule assessment is different, the PFS definition will be hugely impacted. This is really related to your data collection on the CT scan also. And also the change of the healthcare, your baseline characteristics of the patients, their diagnosis will be very different, shifted due to the advancement of the healthcare. So a lot of things we need to take in consideration. So using historical data, thinking about it simple, if we are able to find one, first of all, the source of data, and second of all, how do we deal with all the problems here? So in our paper, we detailed all the questions about that. And for example, choosing high quality data, we even provided a very specific tool, a checklist to evaluate the data standards or quality of data. We also provided suggestion, recommendation, if you design the trial close to the selected source of data, what things you need to pay attention to, such as inclusion, exclusion criteria, or type of design. Like I mentioned already, schedule assessment is part of the study design related efforts, or exactly the definition of your outcome, what kind of potential bias you would consider per your indication of interest related to the time trends of the medical care difference. So all are the things you need to take into consideration. And in the paper, we have a detailed discussion recommendation for each of the topics. And next is a much bigger topic, is about minimize this disadvantage. So for today, I wouldn't have all the time to go over all of them, but 3 big steps in our paper. First is what we talk about on selecting the information, what you pay attention. And second is the design stage, different methodology to minimize the bias of the data. If you back to the step 2. So meta-analysis, causal information, and historical data borrowing categories of different methods. I think as most of the statisticians in this audience, you shouldn't be not familiar with most of them. And our second speaker will also go through some of our explanation and focus on the methodology discussion. So after that, the third step we really want to emphasize is simulation. So after you have the right design, you think you need to choose external data as either historical control or synthetic control, and you choose the right source of the data quality issue is all resolved, you truly can do quite some trial simulation. And also with the selected analysis methodology, as I just mentioned, you can see what really would help to reduce the bias or very different situation to find what's the potential source which impact your trial operating characteristics. And we really truly suggest to find a sweet spot of the power type of error and also reduce the bias and variance. In our paper, we have a very nice graph. I think Rong also will present a little bit where the sweet spot is. We have illustrated in the finding the sweet spots. But with that being said, I'm happy to transition to our next speaker. So 2 speakers in the next. One is Rong, from methodology perspective to discuss. And the last one is Chen Kun. She will really detail a few very important case studies, how to use optimized historical control for approval cases. I think that'll be interesting discussion. Anyway, so Rong, I'll pass the floor to you.
Thanks, Sammy. This very good introduction and give us a good start of the overview of this paper. So for my part, I'll focus a little bit on the methodology review. I think a lot of methodology actually, as Sammy presented earlier, there is a large wealth of the methodology, different approach. But for this part, I will pretty much focus on with a heavy emphasis on the Bayesian approach, but with understanding that there is other things, and such as propensity score and the different approach of doing propensity score in that sense. So in terms of historical data borrowing, and so first it's really not new, right? So it's first proposed by Pocock in 1976. So he mentioned, he lists a few criteria that's when you consider using the historical data, and such as inclusion and exclusion criteria for patient population, type of study design, and exact definition of the outcome. So they are how consistent that's with your current trial, and quality of the study and the potential of the time trend, because of the standard care change, because the and just a spatial effect in the sense. And so most recent development is on the methodology part, can be distinguished on the prior choice between how we choose that. One is a static borrowing, is pre-specified weights and how much we borrow, and that is pre-specified. And the other is dynamic borrowing. The level of borrowing is based on the similarity of historical control with the concurrent data. And this is getting more and more I think encourage signal from the regulatory agencies such as EMA and FDA. 2006 FDA Bayesian statistics in medical device clinical trial, and 2016 FDA extrapolating the existing clinical trial data for pediatrics, and most recently FDA complex innovative trial design. So these are the few guidance that come out that could support the new approach of historical borrowing. And from the experience, what we saw, that's really a lot of things, and especially in phase 2 design, and people are more and more like to try, and I think it's still worth trying. At least at BMS, we are at least giving different trying from different aspect, not only in the phase 2, but also sometimes in the confirmatory trial. So propose historical borrowing, and so I think the best way to really get those efforts out is to keep trying and get feedback and adjust, and eventually, hopefully, I think the trend is it could happen, and when hopefully it's not in the far future, but it's in the near future. And next slides, please, Chen Kun. Based on different approach that says it could be naive pooling, and then the frequentist approach is test then pool, and from Bayesian perspective is a fixed power prior and join the power prior and the conventional power prior, and different, this all belongs to power prior family. And the meta-analytical predictive prior approach and the hierarchical approach, that which is very, in the sense, is very similar to meta-analytical prior. And also mentioned the propensity score matching, and we have also been saying that the application of propensity score matching combined with Bayesian approach, and that's that has been used specific for non-oncology setting. That's the application I have been involved in non-oncology setting, use combining propensity score matching with the Bayesian approach. And so next few slides, I'll focus on the power prior and the other Bayesian approach. So the power prior method was first proposed in 2000, and it assumes historical and concurrent control shares the common parameter, and using the posterior distribution of theta based on the historical data and discount by alpha as the prior for the current control. So this is a kind of fixed borrowing. Basically, is a pre-specified alpha 0 ranging, and that's depending on the how much you want to borrow from, not to borrow at all, 0, up to borrow complete from the historical data up to 1. And the posterior distribution of theta given the historical data D0 and current data D, and the weight of historical data alpha 0 can be specified as giving a likelihood of the prior distribution discount by alpha 0 and incorporate the likelihood of current data. So it will take a so because the alpha is really pre-specified, so it's there's a lot of consideration need to be put into how do you choose the weight and to determine the magnitude of borrowing, and that's the fixed weight power prior. If we go to next slide. And so then people start wondering, can we let data itself to figure out the weight rather than pre-specify? And that's come to joint power prior. And so the modified joint power prior came out in 2015 and treat alpha 0 as an additional random variable and put the beta prior on the alpha 0. And typically beta 1 is first thing that people like to try, and modify the joint power prior to the product of the normalized conditional power prior. This normalization is really making the power prior is a proper distribution, and independent proper power for alpha 0 producing the modified power priors. So as on the formula that you can see that basically is normalized by the conditional power prior, and also based on the likelihood of the posterior of the historical data, like giving D0, which is the historical data. This is the format of power prior. And then next slides, please. So there is another school of thought is commensurate power prior. So this also belongs to power prior family. What is different is it extends the joint power prior to the commensurate power prior, where distribution of a parameter of the current control is centered on the corresponding parameter of historical data. As you can see on the blue part, that current control is centered around the theta hat, theta h and tau, where the tau is the parameter measures the commensurability between the historical data and the current control, which is guided to this tau is really guide the prior on the alpha. Basically, that's provide guidance on the alpha distribution. Next slide. For the meta-analytical predict prior is a historical information can be summarized in a prior distribution, in the sense that for the prior distribution, it will summarize it to become a map prior. As you can see on the middle of the slides, actually according to the Bayes theorem, right? So if the D1 is the current data, and that's the P D1 given theta is the likelihood, and the prior is the map prior. So basically, we use the historical data to formulate the prior, and using that really just if you think in a simplistic way, is in the format of Bayes theorem. And the next slide, please. And the historical model is using the assume the theta h and the theta c are exchangeable following the same distribution. Likely to view that is like using binomial as example. So pH historical data given for the distribution of the historical data, YH is a binomial given NH and pH. And for the current control is similar format, but given is NC and sample size NC and PC. But PC and the pH are coming from is really considered a random draw from the common distribution, which if using if we're talking about logit scale, that's coming from the normal distribution mu theta and sigma 8 square. So basically, they are random draws from a common distribution. That's the hierarchical. It's very analogous to the hierarchical model. And so this historical hierarchical model actually is also known as meta-analytical combined approach, which is called MAC. And so the connection between MAP approach and the MAC approach is that for the MAP approach, is a prior derived from historical data, and it's combining the current control data at the end of the current trial. But for the MAC, which we have is a hierarchical model on this approach. Basically, this approach combine all the control data in one step, right? So basically, it's one single model just with random effect. So I think the really people talking about hierarchical model MAC approach versus MAP approach, they are very similar. Is estimation process differ, but they are very, very similar. So next slide, please. So we listed and went over different approach, but by the end, the big question is how should I choose those approach, and how do I evaluate the performance of those approach? I think simulation study are needed to evaluate operating characteristics such as type 1 error and power. And I think the one of the nice part from this working group is that we have this we propose this this graph this evaluation in a graph format, and so you can see the true historical control, and then you be able to evaluate how the power increase if the historical control where it is different from the current control. And so you can if I also later on we also present this kind of format in a slightly different ways. So for example, on the x-axis, you can use current control rather than the historical data, because then you can mark the historical data on the plot, and then you can see how the current control deviate from historical data, and then how the power change, also how the type 1 error will be affected. And based on this graph, then you can see, okay, so if my historical data is within this range, is not that different from the current control within this part, and then the power, I will have the power gain, but I will not lose too much in terms of type 1 error, or vice versa. So basically, this is always a trade-off balance, and then you can try to find a sweet spot that you feel comfortable to borrow without inflate too much power, inflate too much type 1 error, and without lose power. And also another plot that can be used is mean square error and effective sample size. That really help. In fact, people have controversial about effective sample size, but it's just good that. Yeah, so I'll come to the question. So it's as effective sample size and mean square error, that will help you to give you a better sense of the performance rather than in statistical power and type error. And so I think the performance evaluation is really a key for choosing different methods. So there is no universal one method that we recommend can be applied to all the different situation. And I also want to give a shout out that for my group, and at this year's BEST conference will provide a short course on the different approach for the methodology, and that's a 3-hour short course, and it will get into the detail. Each of the method also provide hands-on experience. Because how to use that, that's a lot of details involved in this, and that short course will provide that is in the October 27th. That people, if you want to get into a little more, and you can send me message. I'll give you the information to register for that course. But that's yes. So you'll take virtual. I think I missed one more. Yeah. So that course will take it virtual. So these are the next slides that Chen Kun will taking over to give a few examples on the real case study that we published in the paper. Thank you very much. We'll come back to other questions later, but I'll hand over to Chen Kun.
Thanks, Sammy, for the great overview, and thank Long for the deep dive on the methodology part. So in the next few sections, I'm going to lead the discussion on case studies on behalf of the team. Before we jump into the real case studies, we want to mention that all material in the following sections were collected from publications, FDA submission packages. Those can be searchable online, but we are not involved in those case studies. There may be some details or rationales behind the scene that only people who heavily involved in those projects may know the answer, but we don't. Also, FDA comments quoted here are available at FDA domain. So let's start with our first case study on ultra-rare disease. It's ultra-rare disease, so the prevalence for that disease is extremely low. Only about 20 patients were reported in the medical literature, and only 15 also have been documented in sufficient details. The disease is called hereditary orthotic aciduria, HOA in short. In this disease, the body cannot make urine dye, which is necessary for life. There are two types of studies included in their NDA. One is a review of medical literature describing urine dye treatment in HOA patients, and the other one is a clinical trial. It's a baseline control single-arm two-center clinical trial conducted in patients with HOA. So this is ultra-rare disease. The prevalence is very low. They are able to include only 4 patients. The analysis for this study is very straightforward. Patient narrative for the medical literature and descriptive analysis for the clinical trials. The comments from FDA indicate that the entire database, that's the 4 patients from clinical trial and the patient from the medical literature, actually represent all confirmed cases of patients with HOA. So with the whole population at hand, they thought the drug effect is adequately characterized and can be generalized into future population for people diagnosed with HOA. And the type of historical control is to compare with the natural history of the disease, with the treatment, without the treatment, the normalization of the endpoint is not expected to occur. So that's the first case study. In the second case study, we will see a study using electronic health records. That's the real world data. A quick summary about the disease and the drug. The disease is called urothelial carcinoma, which is the most common type of bladder cancer. It caused approximately 200,000 deaths worldwide in 2018, and it's the 6th most common type of cancer in US. And among those patients, it's about 1/5 patient have the FI mutation. The drug was approved for patients with urothelial carcinoma, and the tumor have FGFI mutation, and the cancer has spread. Common therapy did not work or is no longer working. So basically, it's advanced bladder cancer. Several studies included in the NDA that include a primary study to support efficacy and safety, and also they provide supportive safety and efficacy data obtaining from additional dosing cohort. There are 2 phase 1 studies to support the safety as well. The study of interest today is one of the RWE study. It's called Flatiron Foundation Medicine Study. Before we go to the details about the RWE study, let's understand the clinical development first. So subjects with advanced bladder cancer was screened for FGFI mutation, and subjects were then randomized into 2 drug regimens. After interim analysis, only 1 regimen stayed, and after the amendment, subjects were placed on the 3rd regimen. That's the primary study to support efficacy and safety. The Flatiron Foundation Medicine Study, the RWE study, is to compare overall survival between a clinical study and the real-world control patient from observational data. So it's easy to tell that the primary endpoint is overall survival, and 1 arm is from clinical trial, and 1 arm is from such observational study. So where are the data coming from? The source of external control for this particular study is like this. At the Flatiron Health Institute, they collect electronic health records from US community-based oncology clinic. So they have real-world longitudinal de-identified patient-level clinic data. And the Flatiron Health collaborate with Foundation Medicine. Their electronic health records are linked with Foundation One next generation sequence. So together, the data set is called Flatiron FMA Clinical Genomic Database. So with electronic health records, they can identify patients with confirmed diagnosis of advanced bladder cancer. And with the next generation sequence, they can provide the testing results for FGFI mutations. Of course, we all know that randomized clinical trial is the gold standard. There are inherited disadvantages if using historical control. So for this particular study, what are the disadvantages and what are the strategy to minimize those advantages? Selection bias. So 1 arm, that's the exposure arm, were from clinical trials, and those clinical trials were conducted in the academic medical centers. However, the control arm were from community oncology clinics. So in general, clinical practice, frequency of patient visit, and the capability of dealing with emergency could be quite different between medical centers and small oncology clinics. So generally, better survival outcome are expected in the medical centers. Also, due to significant missingness, there are discrepancy of inclusion-exclusion criteria between exposure cohort and the control cohort. For example, the ACOG score is a score to quantify the cancer patient's well-being and their activity of daily life. The patient with ACOG score larger than 2 were excluded in the exposure cohort, but those sicker patients were retained in the control cohort. And also more stringent exclusion criteria were observed in the exposure cohort. In general, exposure cohort tend to be healthier than the control cohort. So the recommendation from agency is ask a sponsor to link with other data source, such as claim data, to capture more complete information about the patient medical history and exposure, so that they can improve the capability of applying inclusion-exclusion criteria to the control, so that they can be very similar to the exposure cohort. Another selection bias is from the geographic difference. In the exposure cohort, there are 21% of the patient were from US. However, in the control cohort, 100 patients were in the US. The recommendation from agency is to ask a sponsor to investigate potential influence of geographic heterogeneity on the treatment effect, because people believe that heterogeneity in treatment exists due to different population, different clinical practice in different countries. And they also request the sponsor to conduct a sensitivity analysis to restrict exposure cohort to US patient only. The measure of exposure and outcome can also induce bias. For example, the index date may not be the true first date of therapy in the control cohort. The definition of index date is very straightforward for clinical trials, and we have confidence that it will be the true first dose date of treatment. However, for the real world data, because of the data limitation, the true first dose date of the therapy could be outside of current data set. Again, the agency suggests to link with other data source, such as claim data, to capture more complete information and to increase the accuracy of index date. Also, for this RWE study, the survival outcome is the primary outcome. So it's very important to get the death information. However, how the information on death was obtained in the control were not described clearly. So FDA requests the sponsor to describe how deaths were obtained for the control cohort, whether external data set like the National Death Index were linked to retrieve the death information. Move on to covariate and magic confounders. It's another common area that could introduce the bias. For example, some key adverse prognostic factors distributed differently between exposure cohort and control cohort, but those were not accounted in the study, like the ACOG score we mentioned earlier or baseline comorbidity. Also, there are other unmeasurable potential confounders like the patient race, BMI, socioeconomic status. Because the two cohorts were conducted at different time periods, so that it could introduce temporal bias. Like the exposure cohort were conducted in the early stage of the control cohort. The control cohort have more wider time range. The suggestion from agency is again to link with other data source, such as claim data, to capture more complete information, so that they can improve the determination of baseline comorbidity and other relevant covariant. Also, they suggest to consider the potential confounding factors in the analysis stage if they are not accounted in the design stage. Lack of representative is another issue. As we mentioned earlier, for this case study, blood cancer, such blood cancer is the 6th common type of cancer in US, and about 1/5 patient have such FGFR mutation. If we do the simple math, it can be estimated that there are 140,000 patients have such disease. However, only 25 patients were identified in the control cohort for the primary objective. The population were too small to represent the target population. So the results may not be may not good to generate to larger population. Again, the recommendation from FDA is to get additional data, and it is very necessary to identify control cohort with adequate sample size. So the sponsor has done several statistical models to minimize the bias in the analysis stage. For example, Cox proportional has the model adjusted for left truncation. Also, propensity score approach with different weighting schema. There are some suggestions from agency. They think in general, if there is overlap in propensity score, the two groups are incomparable, and the statistical strategy cannot overcome this problem. So they suggest to provide the distribution of propensity score overlap between the exposure cohort and the control. For this case study, there are something we can learn. In our manuscript, we have a summary of recommendation. Here, I just want to highlight a few. For example, obtain historical control from different sources, like to capture the heterogeneity of treatment and the disease in different population. Also, a well-thought design and proper selection of historical control may not be able to eliminate all the biases. So it's very necessary to adjust for biases using Bayesian or frequency methodologies in the analysis stage. It's good to plan for intensive sensitivity analysis to demonstrate the robustness of results and to determine the weakness or strength of the analysis by varying assumptions. More recommendation can be found in our manuscript. So that concludes the second case study. I will quickly go through the third case study. In the last one, we will see several drugs approved in the same therapeutic areas using different approaches.
Hi, Chen-Kun, maybe you can try to finish this case study by maybe 2 to 3 minutes, so we can have some time for Q&A.
Okay, sure. Sure. So the area is called spinal muscular atrophy. It's a progressive neurodegenerative disease that affects the motor nervous cells. There are 4 primary types of SMA, from type 1 to type 4. They classified based on the onset of the disease from infant onset to the adult onset. And in this area, there are well-established natural history studies. The natural history study focuses on type 1 SMA, which means we have high quality historical data available. And so far, there are 3 drugs approved in this area. Let's look at them one by one. The first one is called Spinraza. It's the first drug approved by FDA. It's an injection administered directly into the central nervous system. So, the sponsor was proposed to use historical control to combine with phase 2 open label dose ranging study. However, it was turned down by FDA because of the confounding factors, like more intensive support care or difference in the patient population. So the drug was eventually approved based on interim efficacy data from a double-blind sham control phase 3 study. The second one is Zolgensma. It's a gene therapy. It's targeted for patients with SMA less than 2-year-old. At that time, Spinraza was already approved, so there are several options in terms of the clinical development. Option 1 is to consider Spinraza as an active concurrent control. It was also recommended by FDA. However, FDA acknowledged that this approach would be very difficult because of the need for repeated invasive procedure. The second option is to use Spinraza as a historical control, but FDA reviewer does not feel that a clear comparison can be made between Spinraza trial and the Zolgensma trial. The third option is to use historical is to use natural history study as a historical control. This is the choice they eventually go. It was acknowledged by FDA that the use of external historical control not ideal, but reasonable in this case because of the unmet medical need and the disease is well characterized. So the drug was eventually approved based on single-arm historical control study. The third one is the first oral treatment for SMA. There are 2 studies. One is called the FIREFISH study. They enroll patients with type 1 SMA. So remember, the natural history study focused on type 1 SMA. So the study design is an open label historical control study. And they want to get broader indication, so they enroll patients with SMA type 2 and type 3 for their second study. And the study design for that study is randomized double-blind placebo control study. So here is a quick summary for this case study. In the same therapeutic area, we saw 3 drugs approved using different approaches. They targeted for different population. The drug administration is different. Clinical development is different. So it will be very helpful and practical to have a checklist to say whether historical control is a good fit for that particular study. So in our manuscript, we provide a detailed checklist. For example, whether the disease is well documented and predictable, whether the endpoint can be objectively measured. So in the end, on behalf of the team, I want to highlight a few points. This is an evolving area, and both pharmaceutical and regulatory bodies are more open to use historical control. And we can try to minimize the biases in the design stage or in the analysis stage. And our working group make effort to lay out the roadmap. And in the end, we want to advertise that currently we are exploring new areas, and those new areas draw much attention in recent years, like the decentralized clinical trial gene therapy. If you are interested, please feel free to contact any of us. So that closes my case study. I will give the floor to the other speakers as well.
Thanks a lot, Shenquan. So wonderful presentation. So I think we have some question in the chat box. I think Ron, thanks a lot for answering the question on true historical control. I think that's been all set for that question. And I think there's another question from Louis, right? I'm not sure Louis is still with us or not. I think he had one question on the terminology, the real world, right? When you present the case study with Flatiron, so he mentioned we shouldn't use the term called the real world. So maybe Louis, if you are still with us, could you elaborate more on what you mean on that? So you can unmute yourself if you want.
Yes, absolutely. Can you hear me?
Thank you. Sure.
Yes. Hi, this is Luis. I completely disagree with the use of the term real world, because especially in rare diseases, when you have only 4 or 6 patients, this basically an extreme good representation, and those patients go through a clinical trial, that data is as real as the non-concurrent data captured in any other source outside of a clinical trial. So I don't like the term real world, because any data is real. So the fact that it's a non-concurrent or capture away from a clinical trial doesn't make it real. It's just non-clinical data captured in a non-clinical trial, and they use it as a non-concurrent, or use the terminology that is already described in ICH E9 is more appropriate than the commercial term real world, real evidence, or whatsoever they do. Okay, thank you.
Thank you.
Yeah, so if other audience if you have any question, you can unmute yourself. But before that, I do have some questions for all the 3 wonderful speakers. The first one is, do you have any general recommendation on the methodology? Ron, you presented several methodology. Is there any general recommendation? For example, you represent the static borrowing, dynamic borrowing. Whether the dynamic borrowing is always better than the static borrowing, or is there any general recommendation on the methodology?
Yeah, I hope to mute myself. Yeah, okay. So I think the methodology that we have been evaluate every time this is a valid question, but we hope to have a solid question for you. Like this is the one we should use. I think it's very, very hard. So depending on, because your current control data will look at very different from trial to trial, and the endpoint will vary. And in general, in theory, people don't like the fixed power prior. They want to rather use the data to inform the weight. And but sometimes that doesn't necessarily guarantee always better, because it's still uncertainty, a lot of uncertainty while you try to estimate that parameter. And so I really so I don't really have a good answer, like say one answer, but I think the simulation really should help us to understand, because anyway we should do the simulation to understand the performance. And because if it's vary too much from the historical data, it's from the clinical data, that you probably don't want to borrow in that case. But how far is too far, and then you need to do the simulation to get a sense of that. I think I don't see a way avoid the simulation have a one-size-fits-all answer. And even for the test and pool, and we found sometimes test and pool is not bad at all. And So it's really I think it's a matter of evaluate too, because this is not that straightforward, and I think when we implement this, a careful evaluation is necessary.
Yeah, Jiayang, that's a good question, actually. I think as exactly as Dong mentioned, during the working group, people have different debate even because from different company, different academic center, and also even different indication of the disease. Some of us have a different preference. So I'm not gonna say which is our preference, each of us. I kind of like, what Rong is saying. Sometimes the simpler one, like system pool, might just work. But that's exactly why in the recommendation we have in the end is you really need to do simulation, and some of them might all work, but maybe the simpler one could be the one. And a lot of people still use propensity score matching. That might still work as the easiest one, especially for rare disease. Actually, the number of people you have might not be necessary to play a very complicated statistic methodology. So it really depends on the situation.
Yeah, I agree. So it's case by case. Like it's not one like methodology fit all studies. I mean, I agree, but just want to know your opinion because you guys are expert in this area. So my another question is regarding the pre-specification of the historical borrowing. So it's more like I at least the case study 1, 2. I'm not sure whether those methodology or what data to borrow, how to borrow was pre-specified before you conduct the study. So to me, like if we want to register or if the trial is for like a registration, right? So the pre-specification is very important. Before you conduct the analysis or even before the study starts, you need to get agreement with the agency in order to like get their like acceptance of your methodology, what data to borrow, how to borrow. Do you have any experience or like a recommendation? For example, if we want to design a new registration study, how would you recommend us to interact with agency? Hopefully I make my question clear. So
yeah, I think the question is clear. So I don't know whether Chen Kun want to take on this one, but to me overall, as I also mentioned in the opening of the discussion, right? If it's really related to regulatory registration trial and you try to use single arm trial for that purpose and use historical control, then you really have a close discussion with agency, what's exactly your plan is. You can see in some of Chen Kun's cases, right? Agency come back and forth with different proposals, looking at maybe your information, your source data, which you plan to use, not enough, asking you to use additional claims data, or even the population is not close enough, they will have a different suggestion. Even methodology, sometimes based on the question, they will have different suggestion. That's exactly why I asked Chen Kun to present a little bit detail on the case study, so the audience and people can see the different situation, what different perspective agency might question you or suggest you. Overall, I think the pre-plan adds all the things you mentioned. If you can pre-plan, you should for sure. But most ended up to is the agency discussion fit for your indication also. So Chen Kun, do you have any other suggestion from the case study review?
Yeah, so thank you, Sammy. I just want to mention that there was another case study that I'm not presented today. In that case study, the sponsor kind of interacted with the agency at a very late stage, and it was criticized by agency.
If I can add just a little bit, right? So I think the pre-specification specification is the key, and how do you making sure you don't choose the method based on what you, not based on what you saw, and the firewall is also important, and make the data, especially when you use the control real world evidence control, when you do this kind of different matching, different historical borrowing approach. I think how to prevent the decision is making based on the not basing on the observed result from your trial, and you have to be very well documented, and how to build the firewall within the company, and making sure that's not only you do it, but also you have to show the agency that you really did it. And so these are critical that to make sure that's taken care of.
Completely agree. Same situation here.
Hi, Luis, do you want to say something?
No. I was saying that thank you, Ron, for that answer. I think that you are totally correct. It is a gamble when you face the agency.
Yeah,
So,
exactly. But that's very helpful discussion, you know, people in practice, how to use the external data in the practice, especially people are mostly trying to see how we can get a product approved. But thank you.
no, and Sammy, something that you mentioned is also critical, because sometimes we have such of a small sample size, and it's not worth it to get so complicated. So the methodology is not a one-on-one thing that you can choose. Sometimes go to the basic.
That's exactly my point, because I work with my department, also work with epidemiology department and other department. I have been looking at so many different case studies, or even internally, and I think some of the simple ways of doing just reference of matching, it works. Why not? Most important is what's the right source of data, whether it works for your indication. Then the complexity of a statistic methodology, to me, it is important if you have those situations, but it's really something we need to think strategically what works, rather than just deep dive into each method and doesn't fit for the purpose. Not necessary. If you look at epidemiology and real world, whatever the department, new department in different industry company they have, they're doing something fit for purpose and really work.
Yeah, and sometimes the biggest challenge is the quality of the data that you have available. So if the quality is all messed up, wow, you just do your best and pray.
Yeah, agree. And actually, we have another paper is coming out soon, and with one of my colleagues internally, it's about how to define utilized time to event data in this situation, because it really matters when the different scheduled visits are different, and how do you adjust that? That could dive into more statistic methodology in depth for sure. And that actually is very important because related to your maybe some of the indication primary endpoints.
Yeah, I mean, thanks a lot, everyone. I think we are almost 10 minutes over our scheduled time. So thanks a lot for joining us. And also thanks again for all the three speakers. If you have any question, you can always reach out to us after the meeting today. And it's Friday. Happy have a good weekend, everyone. So hopefully we can see more works from the working group, the DIA working group, and can present us in the near future. Thanks a lot, everyone.
Thank you, John, organizing. Thanks, Christine. Everyone, bye, guys.
Thank you, everybody. Thank you so much. Excellent.
Thanks. Appreciate
Thank you for
that.
sharing.
And
Bye-bye.
the recording of the event will be available through our YouTube channel. So take care.
Okay,
Thank you.
Christine. Bye-bye.